Hi, good afternoon, everyone, and thank you so much to the CFRI for inviting me to speak. My apologies for not being able to be there physically. And I just want to thank them again for the support for our work. You've heard a lot about acidity and luminal pH all day from this morning. I heard Zach speak, so this will be a little bit familiar to you. I'm going to talk about something old and new, and it's about these CFTR high expressor cells that I discovered many, many years ago, which are of interest to many people in the field of intestinal research now. Um, and what we are learning about these cells and their importance uh, potentially for CF pathogenesis. So when I was a fellow at Yale University, I came to the field just at the time after the CFTR gene was cloned. And my project was to localize CFTR in the intestine using an antibody that was generated by my mentor at the time and was the gold standard CFTR antibody in the field. And so what we did was localize CFTR in the human intestine, and I'm going to show you images from that paid publication. And what we found was that there were these cells located on the villi. You can see one here and another one here, scattered on the villi with really high levels of CFTR. These cells were also located in the superficial crypts. You can see three of them here. And when you look at a lower power image, you can see that they're scattered along the edges of the villi in the small intestine. And at higher power magnification, you can see that there's a lot of CFTR in the apical domain of the cell, but also throughout the cytoplasm of the cell. And that the cells were long and extending from the lumen all the way to the lamina propria. Here you can see a goblet cell that is negatively stained for CFTR. Now, this was important because at the time when I entered the field, the prevailing view was that CFTR is a membrane, it's a channel, of course, and it was restricted to the membrane. And this was completely novel in terms of what was understood at the time. We subsequently looked at the distribution of CFTR in the rat intestine, and here you can see on these panels the distribution of CFTR using the same CFTR antibody that's shown in green fluorescence along the proximal distal length of the rat intestine. And in the proximal duodenum, you can see high levels of CFTR in the Brunner glands, that's at the base, and the apical domain, and in the crypt on the apical domain, and the CFTR decorates the apical domain of the villus epithelial cells, and then these cells with really high levels of CFTR are again seen facing the lumen of the intestine. And these cells are present in the duodenum, they're also in the jejunum, you can see several of them here, and the rest of the intestine you can see that says CFTR decorating the apical domain of the crypts. These cells are absent in the ileum, the proximal, and the distal colon, where you can see CFTR only in the crypts. And I then went on, those are published data. I then went on to look in the mouse intestine, and what we found was that the CFTR distribution in the mouse intestine is similar to that in the rat, except for these cells, that these cells were absent in the mouse intestine. And those observations were published. So then I named these cells CFTR high expressor cells because of the high levels of CFTR. And while I was still a fellow, I went to the Netherlands and did cryo-EM electron microscopy to quantify the subcellular distribution of CFTR in these cells, because based on the fluorescence localization, we hypothesized that perhaps CFTR was trafficking in these cells. And we had to show that, in fact, it was not restricted to the membrane, but it was also within vesicles in the cell. And this was the technique I used. And here you can see an image from rat jejunum showing that by EM, that these cells are electron dense, they are long, and they extend with a basal nucleus. And the brush border is a little bit disrupted on the villus epithelium in these cells. And higher magnification showing the distribution of CFTR by immunogold. You can see the CFTR staining on the microvillar membranes, and then on endosomes, on the membranes of endosomes within the cytoplasm of the cell. Now, the quantification that we did, I'm not showing you that, but that's published. Most of the CFTR is in the cytoplasm. A third of the CFTR is on the membrane, but the majority is in the cytoplasm. And that distribution is one that supported membrane trafficking of CFTR. So we also did immunofluorescence staining of the cells in the superficial crypt. In another publication here, you can see high levels of CFTR in the apical cytoplasm of the cell. And on the basal membrane is the localization of NKCC1, which is an importer, a chloride importer, which is a sodium potassium 2 chloride co-transporter that is critical for importation of chloride into the cell. And CFTR is the apical exit pathway for chloride into the lumen of the intestine. We also looked at the distribution in the rat intestine. You can see here that most of these cells are in the proximal small intestine, and the highest abundance of these cells is actually in the proximal jejunum, shown graphically here, and also in this image where you can see lots of these cells in the proximal jejunum, but they're also in the duodenum, but they're absent in the ileum and in the colon. And these are published data. So fast forward in 2021, when single cell RNA-seq became available, this was an important technological advance in science that allowed elucidation of individual cellular heterogeneity in the intestine. And Hans Clevers was the first to publish single cell data in the human intestine. And so in this paper in 2021, he identified my cells, the CFTR high expressor cells, in the human duodenum by single cell, and he then named them BEST4 positive CFTR high expressor cells, because what he found was that by single cell RNA-seq data, that these cells express very high levels of this gene called bestrophin 4. And the image shown here is that CFTR co-localizes with bestrophin 4 by RNA. And at higher magnification, you can see, just as I had published in 1995, that these BEST4 positive cells are in the superficial crypt and also on the villi. And here you can see a BEST4 positive CFTR positive cells in the crypt. And on the villi, he also found that these cells express very high levels of gluca2b. And gluca2b is the gene that encodes uroguanilin, that's a very important regulator of bicarbonate secretion in acidic portions of the intestine, and that bicarbonate secretion is mediated by CFTR. So here is the co-localization of gluca2b and CFTR in these BEST4 positive cells in the human villar epithelium. So there have been several papers published since then, several single cell RNA papers in human intestine, in other species that have then characterized these BEST4 positive cells. So this comes from a review that was published earlier this year on BEST4 positive cells in the intestinal epithelium. These BEST4 positive cells exist all throughout the gastrointestinal epithelial tract, in the proximal and distal intestine, and in several different species, including monkeys, pigs, rabbits, fish, but they're not in mice. Similar to what I published before about the CFTR high expressor cells. What is interesting is that the BEST4 positive cells in the colon by single cell express very high levels of bestrophin 4, but OTOP2, which is a proton channel, and carbonic anhydrase. These are genes that are important in acid sensing and acid transport, proton transport, and bicarbonate transport. But in the small intestine, these BEST4 positive cells express CFTR, but only in the small intestine. In the large intestine, they do not express CFTR. And in terms of their specification, what's been shown is that these cells arise from absorptive progenitors and that are also regulated by Notch. So Notch regulates cells that become absorptive epithelial cells or secretory cells, which include goblet, enteroendocrine cells, Paneth cells, and Tuff cells, by turning Notch on and off. And the secretory progenitors give rise to goblet and secretory Paneth and Tuff cells and enteroendocrine cells. But what is interesting is that these BEST4 positive cells in the colon are absorptive, as shown in these paper that was published previously. And by localization, you can see in the colonic epithelium, these BEST4 positive cells express OTOP2, the proton channel, and bestrophin 4, and they are in the absorptive part of the epithelium. However, we have shown in previous papers that the cheese cells in the small intestine are functionally secretory, that they respond to secretory agonists by robustly trafficking CFTR to the brush border. And there's also data published showing that the human cheese arise from secretory progenitors. And we now have data that we just submitted in a paper for publication together with my collaborators in the Department of Genetics at Yale, that the rat cheese cells in the small intestine indeed arise from secretory progenitors and involves reactivation of Notch signaling. And we've identified a common progenitor that is shared between cheese cells, Tuff cells, and enteroendocrine cells. So this is very interesting in terms of you have a secretory cell, a predominantly secretory cell that is sitting in an absorptive epithelium. And the new data supports my old published data that here in the human a small intestine on the villi, you can see these cells present, but they lack absorptive hydrolases on the brush border. Hydrolases, both sucrase and lactase, and we know that the villus epithelium is absorptive because they express absorptive hydrolases, except for these cheese cells. In addition to that, morphologically, we showed in 1995 in that gastro paper that these cells have a less well-developed brush border. Here's a cheese cell labeled with CFTR antibody. The cells have a lot of mitochondria and a lot of vesicles, which allow trafficking. And in other papers, we showed that in these cells, they lack a myosin that is critical for the development of the brush border in villus enterocytes. So all villus enterocytes in the epithelium, in developed enterocytes, they express myosin 1A, which is important for the development of the brush border, except for the cheese cells, which lack expression of myosin 1A, which is shown in green. And here you can see a high expressor cell lacking, in the merged image, lacking expression for myosin 1A. So the interesting thing is these cells do not express myosin 1A, and myosin 1A we showed is important for the trafficking of CFTR into the brush border. But these cells traffic CFTR robustly, but they lack myosin 1A. And we still did not understand which motor was regulating the trafficking of CFTR in these cells. So one interesting question then in the field of BEST4 in terms of the intestine is BEST4 positive cells are identified along the intestinal tract, but there's only CFTR present in the cells that are in the proximal small intestine. And why is that? And so we believe, and we're hypothesizing, that that is linked to the pathophysiology of CF intestinal disease. Luminal acidity is an important pathogenetic factor in gut disease and cystic fibrosis. So, as you heard by others today, that gastric acid comes into the small intestine where it is buffered by bicarbonate, and that bicarbonate secretion is mediated by CFTR. But in the cystic fibrosis intestine, it is the pH is much lower, and that lower pH is a result of the lack of functional CFTR in the enterocytes. And the lack of that CFTR and the lack of bicarbonate secretion leads to increased acidity that contributes to the development of thickened mucus, intestinal obstruction, bacterial overgrowth, gut inflammation, malnutrition, and also gives an increased risk for GI cancers, which we now know is more present in patients with cystic fibrosis. So, in terms of the small intestine and CFTR, there's a critical role for chloride and bicarbonate secretion. And in the small intestine, where you have villi, here's a diagram of the cells along the crypt-villus axis, and their highest levels of CFTR in the crypt, and the levels of CFTR are lower in the villi, except for the high expressor cells, which are shown in bright blue. Here you can see an enteroendocrine cell, goblet cells, and transit amplifying cells, Paneth cells which secrete antimicrobial peptides. And in terms of CFTR, what we showed before is you can see higher levels of CFTR in the crypt, in the jejunum, and as the cells migrate up the villus, you can see less CFTR except for the high levels of CFTR. Now, others, including Lane Clark and our group, have shown that villus epithelial cells and Zach's group secrete bicarbonate through CFTR. So it's an important segment of the intestine where you have higher acidity, that CFTR plays a crucial role in that part of the gut because of its important function in bicarbonate secretion. So what are these cells? We have collected a lot of data now to support the notion that these cells are a novel subtype of epithelial cell. Number one, because of their location in the proximal small intestine, and the fact that this portion of the small intestine is more acidic compared to the lower part of the intestine that's more alkaline. These cells also respond to secretagogues, including cyclic AMP, where the CFTR traffics robustly in response to cyclic AMP into the brush border, and by cyclic GMP, which I also published. And when we looked at the single cell RNA data, what we found is that there are lots of genes that are highly upregulated in these cells that are in the cyclic GMP pathway, including what was shown in the human intestine by Hans Clever's gluca2B that encodes for uroguanilin, which regulates bicarbonate secretion in acidic portions of the gut that is mediated by CFTR. The receptor for uroguanilin, which is GCC, otopterin 2, which was shown in the BEST4 positive cells in the colon, which is a proton channel. And interestingly enough, what we found is that these cells are enriched for acid sensing receptors. So GPR4 is an acid sensing receptor. And again, the bestrophin 4. And in terms of transcriptional regulation, in contrast to ionocytes, we found that these cells express very high levels of MICE1, which is a homeobox gene. So in summary, the single cell data is showing us that these cells are enriched in acid sensing machinery genes and cyclic GMP machinery genes that are important in bicarbonate secretion. So ionocytes were identified around the same times that I identified the cheese in the small intestine. And there's a lot more work that's been done in ionocytes. including identification of the transcription factor FOXI1 that regulates ionocytes. So both of these cell types express very high levels of CFTR, but the single cell data reveal very different transcriptomes for ionocytes and the cheese cells in the small intestine. So here you can see our single cell data that the mice one is exclusively expressed in these cheese cells. And when we do protein labeling using antibodies, you can see that we can identify these cheese cells by CFTR staining in green, but by mice one in the nucleus that's restricted to the cheese cells, both on the villi and in the superficial crypt. We also wanted to identify whether FOXI1, which was shown in terms of ionocytes, whether FOXI1 was present in the cheese cells. So we labeled mouse trachea, which has ionocytes, but remember, mice do not have cheese cells, but the mice have ionocytes in the trachea. And we labeled for CFTR using my antibody and FOXI1, and we can identify FOXI1 in ionocytes in mouse trachea, but we could not identify any cheese cells with FOXI1 in the jejunum, or in fact in any part of the intestine. So FOXI1 is not in the intestine, and it does not localize to cheese cells, whereas FOXI1 is important for ionocytes in the lung. So what about these cells, and what have we learned about these cells in terms of the single cell? What we've learned is that these cells are enriched in genes and proteins that regulate brush border membrane trafficking, the pH-sensing, and cyclic GMP activated bicarbonate secretion. And this I showed you in the previous slide before. So as I showed you before, myosin 1A is absent in these cells, but cells traffic robustly. So the single cell data identified myosin 1B, and here you can see localization of myosin 1B protein in a cheese cell identified by mice 1. And here you can see 2 cheese cells labeled with myosin 1B and CFTR, identifying that this myosin is exquisitely expressed in the cheese cell. In terms of the other genes that we identified, bestrophin 4, we saw, you can see the basolateral membrane localization of bestrophin 4 at the protein level, and there's a basal process in these cells beneath the nucleus that's extending into the lamina propria. There's also high levels of TUB2, which is expected. This is tubulin, which is important in trafficking, and we know that these cells traffic robustly. We got antibodies from an investigator to uroguanilin. This is the protein that's encoded by GUCA2B, and you can see very high levels of uroguanilin exquisitely in the cheese cell. And interestingly enough, uroguanilin is also present in these basal processes. GCC, which is the receptor for uroguanilin and for cyclic GMP ligands, is highly expressed in the cheese cells. And this is very interesting because this is a receptor that is predicted to be on the apical membrane. But here you can see we have really high levels of the GCC in the apical cytoplasm and throughout the cytoplasm of the cheese cell. And the bestrophin 4, even though it's a basolateral membrane channel, we find it at the protein level, both on the basolateral domain and what looks like vesicles in the cheese cells, suggesting that these bestrophin 4 may also be important in trafficking within these cheese cells. So together with my colleague in genetics, my collaborator, we developed a rat organoid model to be able to identify and work functionally with these cells. And so we developed a rat jejunal organoid, since these cells are most abundant in the rat jejunum. So here you can see in the undifferentiated cell state of the organoid, you do not see these high expressor cells, but similar to what we find in the native tissue, that in the differentiated organoids, you see several cheese cells. And you can see in this cross section, we can identify them again by mice 1 staining in the nucleus and CFTR, and we identified bestrophin 4. And this was published in a publication last year in June, the rat jejunal enteroid model. So I'm going to talk now about the neuropod cells because that's the title of the talk. And so neuropod cells were identified relatively recently by Boroquez in 2015. And these are enteroendocrine cells that are been identified sporadically in the gut. And they have a specific morphological appearance where there's a brush border of the cell, but the cell has this long basal process with synaptic vesicles at the base that interact with the efferent and afferent nervous network and the enteric nervous system that interacts with the brain. So there's rapid transmission through the synaptic transmission process that allows these cells to sense luminal contents and interact with the nervous networks of the brain and respond to luminal contents in the brain, luminal contents in the intestine. And so there are particular genes that have been identified in neuropod cells. So we looked in our single cell data to see whether we can identify genes in the rat jejunum that would meet the criteria for neuropod cells. And what we found that is, in fact, we did. TUB3, synaptotagmin 3, neurofilament medium, S100A6, choline acetyltransferase or CHAT, and GCC. Now, GCC was identified in neuropod cells last year in a paper in mouse intestine, in the small intestine. And in those neuropod cells, synapsin 1 was also present, as shown in this image here in red. And here you can see in the merged image, synapsin 1 in red and GCC in green in mouse small intestine. So this GCC is an important neuropod gene. And so we then began to label the chi cells in the rat intestine to see whether we can identify these neuropod proteins in the rat chi cells. And here you can see a chi cell labeled for BES4. So, and you can see a long basal process labeled for BES4 extending into the lamina propria and connecting to TUB3. We identified synaptotagmin 3, similarly in close proximity to a BES4 positive cell. And the neurofilament is also present, but it's also present in the basal portion of enterocytes in the villus epithelium, which suggests that neurofilament is interacting also with the nervous network. The S100A6 is shown in a chi cell here. A lot of prominent, this is a calcium receptor that is present in the apical domain. And we know that CFTR is also regulated by calcium. And this is an important player in neuropod transmission. The CHAT cholinesterase transferase, an important regulator, cholinergic regulator of mucus secretion in goblet cells. And here you can see that the CHAT decorates the lateral membrane of a goblet cell, which is what we expected, but we also saw it in the chi cells. And again, the GCC, we identified the protein throughout the apical and the cytoplasm of a chi cell. So bestrophin 4, which is the signature gene and protein that's identified in these cells, you can see the bestrophin 4 in very close opposition to TUB3, and this video image shows the nice, the close proximity of the BES4 positive cell to TUB3. So the next question was, since these cells behave in a secretory manner, they express acid sensing genes, acid sensing receptors, and the proton channel O-topterin 2, and a lot of the cyclic GMP machinery to regulate bicarbonate secretion. We wondered whether these cells are the sensors of acidic pH in the small intestine. So we then looked at O-topterin 2 in chi cells under normal conditions and under conditions when we lower the pH. And we did this doing physiologic experiments with native intestine, where we lowered the pH in the lumen. And here you can see what was very interesting is that a mice 1 cheese cell, which is labeled in magenta, has no autopterin staining under conditions when the pH is neutral. But when you lower the pH to 2, you can see a robust upregulation of autopterin in the apical portion of the cell. And this is similar to and supports data that's already published about the BEST4 positive cells in the colon by the group that published this in 2019 in Nature, that when they use a rhodo-red dye, which is an indicator of low pH in the cell, when you lower the pH to 5, rhodo-red fluorescence increases in the BEST4 positive cell, and it's shown here graphically. And I'm not showing you this data, but we have new data using the same rhodo-red dye in our cheese cells in the small intestine, confirming that indeed the cheese cells are functionally proton importers. So they're sensing and taking up the protons into the cell, similar to the BEST4 positive cells in the colon. So what about CFTR in the cheese cells in response to low pH? You can see here, we looked at CFTR also. So in the normal condition, there's a lot of apical CFTR, but when you lower the pH, it moves to the apical domain, and that's shown also graphically by the quantification. GCC has a similar kind of response that you can see the GCC in green here, upregulated in the apical portion of the cheese cell when you lower the pH in the lumen. And interestingly enough, when you lower the pH in the lumen of the intestine is able to buffer that low pH and to normalize the pH, as you can see here, that you lower the pH and then it comes back to normal because you have buffering capability. And we believe that that is occurring because of not just the other villus epithelial cells, but because of the cheese cells sensing the pH and then responding to that. Here is published data from my lab, also looking in the duodenum in the rat, that these cheese cells do not express NHE3, which is an important transporter that regulates sodium absorption, but they express high levels of CFTR. When you lower the pH, there's upregulation of CFTR to the brush border of these cells by trafficking. These cells also express very high levels of the vacuolar ATPase, as shown here in red, that co-localizes with CFTR. This is a very important transporter that regulates proton transport into and out of cells. So what about CF? So because these cells are not present in mice, and we do not have very good models, and we do not have a rat CF model, we collaborated with a group in Australia that generated a delta F508 rat model for their studies in the lung. And we worked with them to get samples of intestine from the rat delta F508. And we then used those tissues at Yale to look at in situ hybridization, to look at mRNA levels of CFTR, because really nothing is known about these cells in the human intestine. And more importantly, nothing is known about these cells in the human CF intestine. So if we can learn something about these cells in the rat model, it will really help us. So here you can see at the RNA level, CFTR in a villus cheese cell and in the superficial crypt. We also looked at gluca2b, and you can see that that expression at the RNA level is preserved on the villus and also in the superficial crypt. We looked at delta F508, and you can see on the villus cheese cells, markedly reduced expression of CFTR, and so in the crypt, but the gluca2b expression is preserved in the delta F, both on the villus and in the crypt. And what about the both gluca2b and CFTR as similar to what Hans did in the human? So in the wild type, you can see in the merged image CFTR and gluca2b, both in the duodenum and in the jejunum in these cells, but you can also see gluco-2b, some lower levels of gluco-2b expression in the epithelium. These cells, as I showed before, are absent in the ileum and colon, but you can see gluco-2 positive cells in the colon, in the absorptive epithelium. These are the best 4 positive cells that were shown by others. In the delta F508, you can see CFTR and gluco-2b. There's a lot less CFTR, but the gluco-2b is preserved. In the jejunum, same thing, less CFTR, and there's in the merged image in the ileum and the colon, gluco-2b preserved in the best 4 positive cells in the colon. You could see these cells here, but they are the Chi cells are absent in the ileum and in the colon. And what about CFTR? So here we have images both in the wild type, the delta F508, and the CF knockout rat. And in the wild type, you can see the beautiful Chi cells with a lot of CFTR in the apical domain and the brush border. But in the delta F508, you see much, much reduced CFTR and no CFTR in the knockout, but mice one is preserved in the knockout, right? So this is sort of interesting. And in terms of GCC, we looked at that also. The GCC is preserved, but it appears to be increased in the delta F508. And you can see much higher levels of GCC in the Chi cell in the delta F508. And that is shown graphically here in terms of the fluorescence intensity. And we also noticed that there was an increased abundance of these cells in the delta F508 jejunum, suggesting that there's some compensatory attempt in the delta F508. Now, we don't know what is happening in the human delta F508 intestine because we don't have access to that tissue. But certainly in the rat, there appears to be compensation. So I will conclude by saying that what we've identified is that these Chi cells are neuropod cells, and they appear to sense and respond to changes in luminal pH. We believe that they play a significant role in CF pathogenesis, particularly with regards to the sensing of the pH in that part of the intestine and responding. Where are we going going here forward? We are going to try to look at the functional studies to look and understand these cells further and to understand the role of BEST4 in the Chi cells and also the role of these cells in human gut disease. There are, of course, challenges because of the models. We will begin also by looking at normal human organoids and CF organoids to try to understand a little bit of these cells. And I want to thank the CFRI for supporting us. And this work was primarily conducted by a post-grad from my lab, Jason Jin, who's shown here. He just left to go to medical school, and the other members of the lab, Caroline Mueller and Reginald Streeter, who is the new post-bac, who's joined the lab, my collaborators in the Sumigray lab, and the University of Adelaide in Australia, Peggy Mung at Yale to help us with the RNA studies, and the other support that we received from NIH and CFF to help us to complete some of this work. So I thank you, and I'll be happy to take any questions.